Okay, so today we are going to discuss the physiology of vision, which will start with in the first by the physiology of the retina, and then we will go through the physiology of the visual pathway, like the lateral geniculate body, like the visual cortex. We will know how does the visual signal transmitted or is transmitted through these stations or these areas, and we will discuss everything about them. So I will try. Please give me your undivided attention. And if you need any help or if something is unclear, please let me know in the chat box. Okay, so today or with this lecture, we will give just an introduction about the functional anatomy of the retina, and then we will go through the role of vitamin A in the physiology of vision. Then we will discuss the visual cycle. How does the visual pigment break the process of what's called bleaching or breaking down of the visual pigment and the regeneration. Then after the visual impulse is initiated in the retina, how does it is transmitted through the visual pathway? Let's have first some introduction about the retina. So we know that the retina is a multi-layered sensory tissue that lines the back of the eye that have millions of receptors. What are the functions of these receptors? Well, the role of the retina is that it will convert the light rays from a photochemic or a photons or photopic stimulation into electrical stimulation. So the light itself is not an electrical impulse, but the retina has a nerve, and the nerve, what is conveyed through the nerve is electrical impulses. So the role of the retina is to convert these light rays into electrical impulses, and this is what's called a photochemical reaction. So through series of chemical reactions an electrical current is produced through the retina, and it is transmitted through the optic nerve to brain, where the brain will translate these electrical impulses into images. So this is just simply what is the process of vision. So once a person see an image, this image like is transmitted through photons. These photons are received by the retina, and the retina, these photons will stimulate a series of photochemical reactions through the retina that will convert these photons into electrical impulses. These electrical impulses generate in the retina and will be transmitted through different layers of the retina, as we will see. Then it will be transmitted through the optic nerve to reach the lateral geniculate body, which will be the second order neuron of the visual pathway, and then finally will be received and interpreted by the visual cortex. So just a quick review about the anatomy of the retina, and because this will reflect the physiology, we know that the retina has 10 layers from outer to inner, starting by the retinal pigment epithelium. This is the outer layer, and inner 9 layers, the photoreceptors layers, and the plexiform layers, the outer and inner, the nuclear layer, the outer and inner, and the ganglion cell layer, and the retinal fiber layer, and the inner limiting membrane and external limiting membrane. So these layers are of the retina. Let's see them from outer to inner. It is the RPE photoreceptor layer, the outer limiting membrane, outer nuclear layer, outer plexiform layer, inner nuclear layer, inner plexiform layer, ganglion cell layer, and the nerve fiber layer. So the electrical impulses, you can see that the nerve fiber layer is the most inner one, and the retinal pigment layer is the most outer one. So the strange thing here, but it is not very strange. It has some logic and some sense behind it. That the light, when it is striking the retina, it doesn't strike the photoreceptors in the first instance, but it will go from outside. So it will pass through the vitreous. Here is the retina, and it will pass through the different layers of the retina. Then it will be received by the retinal pigment epithelium, and then the light impulse will be initiated in the retrograde manner. So the light impulse will be initiated from the photoreceptors here, or this is the primary organ for sensing the light. And then electrical impulses will be initiated in the photoreceptors, will be transmitted into the bipolar, and then the horizontal and amacrine, then the ganglion cell, then it will be transmitted to the optic nerve. So this concept should be clear in your mind, that the path of light is the in reverse direction of the pathway of visual processing. So starting by the different important anatomical regions within the retina, we have the fovea. As we know, it is the central depression in the posterior pole of the eye, which will become the most sensitive part of the retina. It's about 1 millimeter square that contain only cones, and as you can see that the other layers like ganglion cells and bipolar cells are displaced to the sides, so that the light will hit the photoreceptors immediately. This will lead to improvement of the visual acuity, or that will lead this anatomical configuration will make the fovea the most sensitive part, and the most and the area with the greatest visual acuity, as the light strikes the cones directly. Another important region for the retina is the pigment layer of the retina, or the retinal pigment epithelium. You remember this figure from the anatomy. This is the retinal pigment epithelium, and we said that it contains melanin. So these black spots are melanin. So what is the role of melanin here? The role of melanin that it will prevent light reflection in the globe. So if it is without this melanin, then when the light will strike the retina, it will undergo scattering. So the scattering of light will impair the visual acuity. So what is the role of melanin here? That when the light is striking the retina, this melanin will absorb the excess light, so that it will prevent it scattering through the eye, and thus it will improve the visual acuity. So you can consider it like your ordinary sunglasses. So why you are wearing dark sunglasses in the daylight? This is because you want to protect your eye from the excess light. This is the same thing for in the RPE for the melanin to the RPE. The melanin will protect the RPE and protect the retina from the excess light. And that's why those who have albino, those who have albinos, or those albinos will be complaining from photophobia and they decrease the visual acuity, because they lack the melanin, so that they have excessive scattering of light within their eyes, and there will be so this will result in poor visual acuity. Also, other functions of the retinal pigment epithelium, as we will see now, it will provide binding sites for free radicals and peroxides. So let's see what are the functions of retinal pigment epithelium. We said the first one, it is the light absorption. So it will it will absorb the scattered light. The second thing, it is a site for epithelial transport. This is because the retinal pigment epithelium form the outer boundary of the blood-retinal barrier. So the blood-retinal barrier is a barrier formed between the retinal pigment epithelium and the non-fenestrated retinal capillaries, so that it will the tight junction between them will prevent the molecules and the excess water to pass freely between the cells. So if, like, for example, if the water is in the body or within the bloodstream, it will be prevented from entering the eye or entering the retina by it has a large molecular size, so that it will be prevented by the blood-retinal barrier to enter the retina. Not only for microorganisms, but for certain molecules and for certain nutrients, and it will control the exchange of nutrients and water from the circulation to the retina. And also it has a function that it will pump excess water between the RPE and the neurosensor retina. So it is not uncommon, for example, for people who undergo retinal detachment surgery and they have the retina detached, and sometimes after the surgery the retina is not completely attached, and there is a separation between the neurosensor retina and the retinal pigment epithelium. But in this occasion, we rely on the RPE pump in order to pump excess water from this potential space and to pump it into the choroidal circulation. The third rule here is the visual cycle. We will have a thorough talk about the vitamin A and its role in the vision. So we will postpone this later, as well as the phagocytosis of the outer segment of the photoreceptor membrane. So we will know in a while. We will talk about the process of vision and the process of visual cycle, and we will say that the outer segment photoreceptors will be destroyed in a continuous manner, so that these outer segments will be destroyed in the process of bleaching, and then will be engulfed by the RPE. So these outer segment engulfed will, or this engulfed RPE will be or this engulfed outer segment will be a source of vitamin A, as we will see later. And this will be due to the exposure of light. Also, it will be responsible for secretion of some materials like a vascular endothelial growth factor, like a platelet endothelial derived factor. These are also one of the functions of the retinal pigment epithelium, and also it has an immune function, either passive or active. So passively, it will provide a barrier, as we said, as a part of the blood-retinal barrier, and active by secretion of some immunological substance like the cytokines. So here is the different barriers of the eye. We have mentioned before about the blood-aqueous barrier. We said that it is a barrier between the tight junction between the lateral surfaces of the non-pigmented ciliary epithelium and the non-fenestrated iris capillaries, and this barrier will control the things that may enter the aqueous. The same thing here with the blood-retinal barrier. This is a barrier between the tight junction between the retinal pigment epithelium, which will be the outer blood-retinal barrier, and the tight junction between the non-fenestrated capillary endothelium of the retina, which will be the inner blood-retinal barrier. So to remember, the blood-retinal barrier is formed between the RP tight junctions and the non-fenestrated retinal vascular endothelium, while the blood-aqueous barrier, it is formed between the non-pigmented ciliary epithelium tight junction and the non-fenestrated iris vascular endothelium. So here is a summary for the functions of retinal pigment epithelium. First of all, it is absorbed the light absorption. So it will absorb the light. The melanin here will cause preventing the scattering of light. It is responsible for epithelial transport. So it will, as it's formed the outer boundary of the blood-retinal barrier, it allow nutrients like glucose and the vitamin A to enter through the RPE coming from the capillary vascular bed or the choroidal vascular bed. Will allow the chloride to enter to diffuse out. It will pump the water from the space between the RPE and the neurosensory retina, so that the blood will not accumulate and the retina will be attached. Also, just to mention some important things about glucose here, the glucose, the RPE, so the RPE will be the most or the highest amount of respiration, or respiratory rate is the highest in the RPE than any other part of the body. It is even twice that of the brain. And the glucose, and the unique thing about the retina here, that the retinal cells or the RP cells doesn't require insulin for the glucose to enter the cells. This will tell you how much these cells are metabolically active, and that they even can possess the glucose without the need of insulin. Also, the other thing that is unique about the retinal cells, that even with the presence of oxygen, the glycolysis, the anaerobic glycolysis, is the main source for energy. So these cells can do anaerobic glycolysis even with the presence of oxygen, unlike other cells. So other cells, if they have like the lens, for example, or the cornea, if there is oxygen, okay, we will go through the Krebs cycle, so through aerobic glycolysis. But if there is no glucose, we will go through anaerobic glycolysis, producing the lactate and the pyruvate. But in the retina can do glycolysis even with the presence of oxygen. This is just a thing about the retinal metabolism. Immune function, that it production of PDEF or platelet endothelial growth factor and vascular endothelial growth factor. the visual cycle. It will be a part, an integral part of the visual cycle, as their 11 cis retinal, which one form of the vitamin A will circulate through these retinal cells, and will be also the RP cells will take the vitamin A or another form of vitamin A from the blood capillaries, and will be having an important role for the visual cycle, as we will see later on. Also, the phagocytosis of the outer segment of photoreceptors, as you can see, the under exposure to light with the oxidative light stress, these photoreceptor outer segments will be destroyed and will be engulfed through the RB cells, and will be reproduced for regeneration of these discs. Secretion of platelet endothelial derived factor, vascular endothelial growth factor, and immune function by the secretion of cytokines for the active, and the passive by prevention of microorganisms from entering the eye by the barrier function. Hello? You cannot see the lecture? So for those who cannot see or cannot hear, they have a problem with their connection. So you need to figure it out and resolve it, since others can see it. Okay, so we'll continue. Another important cell which have an important function of the retina, which is Muller cell. We have talked about Muller cells in the anatomy, and we said that it is a very long cell starting from or extending throughout the whole length of the retina, starting from the inner limiting membrane and the outer limiting membrane, and this is the cell body. So here is the cell body, here is whole the length, this is the external limiting membrane, and here is the internal limiting membrane. So that since it is very long, it will have a supportive function for the retina, and this we have mentioned before. But other functions of the Muller cells that you need to know, that it is a storage form for the glucose and retinoids. This is very important because this cell have a specific feature of possessing an enzyme called glucose 6-phosphate. So you remember this glucose 6-phosphate from the previous lectures in the anatomy, in the physiology of the cornea and lens. We said this enzyme will be able to metabolize glucose, so that this enzyme in the cells will enable them to metabolize glucose into their stores, and it will be making the neuroretina will be able to benefit from these glucose stores. So they have the glucose, they have the enzyme that metabolizes the glucose, so it can produce some energy. Also, as we will say in the electrophysiology, it will be responsible for the B wave of the ERG. So the B wave of the ERG will be responsible by two cells. One of them is Muller, and the other will be the bipolar cells. Okay, so now we talk about the main issue, or the main part, which is responsible for the vision, which will be the photoreceptors. So these are the cells responsible for sensing the light. These are cells which are the rods and the cones. So let's now figure out what are the differences between the rods and the cones. So the rods are the most sensitive to light and dark changes. So since the rods are responsible for mainly in the dark vision, that's why they need to be very, very sensitive, because with the single photon of light, there will initiate visual impulse in these rods. So this is these types of photoreceptors will contain a light sensitive pigment. So we have what is called visual pigments. The visual pigments are a sort of chemicals present in the photoreceptors that are responsible for initiation of the sense of light. The type of visual pigments present in the rods is called the light-sensitive pigments or rhodopsin. The rods are more numerous than the cones and the extending more toward the periphery so that it will be able to collect any light during the dark, so that it will be about 120 million rods in the human retina. Conversely, the cones are not as sensitive to light as the rods, because if the cones are very sensitive and we are seeing by the cones in the light, the cones will be burned out because they are very sensitive. So sensitive means that it will be able to appreciate the least amount of light. And we don't need the cones to be very sensitive. So the cone also has pigments. These pigments are of three types of colors, either green or red or blue. So the cones are mainly responsible for appreciation of the color vision. So they have three types of cones, and each one will be carrying one single type of visual pigment that is responsible for seeing a specific color, either the green or red and blue. And that's why we cannot see the colors very well in the dark places, as the cones work only in the bright light. So for example, if you see this picture, this is while seeing this scenery in the daylight, we can appreciate the blue color, the green color, and different colors because we are seeing in the daylight by the cones, and the cones here can see the colors very well. This is not the case when seeing the same scene in the dark, because in the dark we see only by the rods, and the rods are like a color blind also, or they are not very sensitive to the colors, so that we can see everything in the shades of gray. Let's now see what are the different parts of the photoreceptors. We have also discussed this in the anatomy. Just a quick review in order to know what we are talking about. We have first of all the outer segment of the photoreceptor. This outer segment of the photoreceptor is the part that contains the visual pigment. So in case of the rods, it will be the rhodopsin or the light sensitive pigment, and in case of the cones, it will be the three colored pigments. These light pigments are present in discs. These discs are around 1000 discs, and they are considered like folds of the cell membrane. The photopigment is present as a transmembrane protein, so in every disc, the photopigment will be present here like a transmembrane protein and forming about 40% of the segment mass. Then we have the inner segment, and this inner segment will consist of, will contain cytoplasm, will contain organelles like Golgi apparatus, like mitochondria, which will be the most important part here, as this is the part of energy production. Let's say about 50% of the energy production of the retina will be produced in this part, which will be the inner segment of the photoreceptor, as it contains mitochondria. Then we have the nucleus, and then we have a synaptic body, which can be rod spherule or cone pedicle, which will make the synapse with the bipolar cells. So to see a better picture for what is the junction between the outer segment or the photoreceptor and the retinal pigment epithelium, you can see that there is a microvilli of the outer of the retinal pigment epithelium that contain melanin granules, and the outer segment of the photoreceptor is interdigitated with these microvilli. This has an important role because the light impulse will be initiated here. So these discs will be broken, and they will under the effect of light, as well as the light will cause bleaching of the visual pigments here, and will be engulfed through the retinal pigment epithelium. Here again, a section for showing the structure of rods and cones. You can see that this is the rod showing this is a disc and a disc and a disc and a disc. So this is called the outer segment of the rod. And this is the cone showing the outer segments of the cone, showing also multiple discs. So the visual pigments are present as a transmembrane protein here and there. So to summarize, what are the difference between the rods and cones? They will be different in number, as the rods are much more numerous, much more dispersed through the periphery, while the cones more in the center, as it was present more in the fovea, while the rods are not present in the fovea. The photosensitive pigment in the rod is called the rhodopsin. This is the visual pigment, which is the light sensitive. The cones have different types of photosensitive pigments. They are called iodopsin collectively, but they have other names. But we don't have a specific name for the cones. We say that it is just the visual pigments of the cones. The rods, since they are very numerous, they will show the phenomenon of convergence. So about 300 rods will converge to one bipolar cell, while the cones does not show this phenomenon because they are few, so they doesn't show no convergence. So they will be one cone will convey that the signal to one bipolar cell. So it's like a direct private line. Why do we need to this? I will tell you now. So what are the functions of rods? They are more sensitive, so they will be much more or will work in the during the dim vision or dim illumination, as they are very light sensitive. So once there is a light, they will be able to see it. But on the other hand, they have poor visual acuity. They cannot see the color vision, and they are important for the night vision. The reverse would be the cones. So the cones have decreased light sensitivity because their function is mainly in the daylight, and they will have much better visual acuity because of that there is no convergence, and they will be able to see the color visions by the three types of the photopigments, and it is responsible for the day vision. So here is what we meant by convergence. In case of the rods, since they are 120 million, they show the process of convergence. So about different or numerous or large number of rods will converge to a single bipolar cells, and then the large number of bipolar cells will converge to a single ganglion cell. That will lead to decrease the visual acuity, as the visual process will be concised to a smaller number of bipolar and even more smaller number of ganglion cells. While in case of cones, the single cone will be connected to a single bipolar, connected to a single ganglion cell, so that this process of convergence is not present here. So every cone will lead to what's called point-to-point transmission of visual impulses. So every cone will contain a single visual impulse, will be transmitted to a single bipolar, then transmitted to a single ganglion cells. So the visual processing will be of much more accuracy, leading to improving the visual acuity. So now we have a question to me. We just said something about it in the retina, in the anatomy. Why is the fovea is the most sensitive part of the retina? We said something in the anatomy, which is the anatomical configuration, that all layers are shifted to the side, allowing the light to strike the photoreceptors immediately. So there will be no intervening layers between the photoreceptor and the light. Also, we said that high density of small diameter cones, and they are more long and more slender. And now we have in the physiology, now we will say that it has a one-to-one convergence. So cone will connect to a bipolar cell to the ganglion cell. And we say that there is a wider representation of the fovea in the occipital cortex. So it's like about 1/3 of the occipital cortex is represented by the fovea. So here is what is meant by convergence. Again, the rods will show larger convergence. So that will allowing large areas or large number of rods to transmit the signal to a single bipolar, while the fovea with high density will show smaller convergence, even 1 to 1. So is everything clear up till now? Okay, so we will talk about the vitamin A metabolism and its role in the visual cycle. So we hear always you need to eat a lot of carrots in order to improve your vision, and those who eat carrot will be have healthy vision and a healthy visual acuity. So does this have any scientific basis? That's what we discuss now. So what are vitamins? Vitamins are organic compounds required in diet in small amounts to perform specific biological functions for normal maintenance of optimum growth and health of the organism. So we have different types of vitamins. Some are fat soluble, some are water soluble. So the vitamin A falls under the category of fat soluble vitamins. So what are the sources of vitamin A? So vitamin A is an essential fat-soluble vitamin present in different forms. The first one is called the active form. And the active form is exclusively present in the animal products, like cheese, like liver, like other meat. So these are the sources of active form of vitamin A. By meaning the active form of vitamin A, which will be incorporated into the visual cycle, which are called the retinoids. So retinoids it is a term collectively meaning that these are different forms of vitamin A. This can be retinal or retinol or retinoic acid. These are different metabolic forms for the vitamin A, and all are active forms. Another form is called the provitamin A. The provitamin A an immature form of vitamin A, or it is inactive form of the vitamin A. It is called collectively the carotenoids. So as it is apparent from its name, it is coming from carrots. So this type of vitamin A is present in the plant products like carrots. And this part or this form of vitamin A is not active, so it needs to be activated into retinoid in order to be functioning. And this occurs in the body. Okay, so what are the types of vitamin A? Let's see, we have different types of vitamin A, or different, the first one, as we said, it is the provitamin A, which is called the beta carotene. And we have the retinoids. The retinoids are different in their chemical structures and different in their names. So we have the retinol, and we have the 3-dihydroretinol, and we have the retinal and retinoic acid and retinyl esters. So when it is the vitamin A, it is present in the alcohol form, it is called retinol. Or if it is aldehyde, it is called retinal. And if it is stratified, then it is called retinyl esters. So these are the main forms of the vitamin A that we will talk about in the metabolism of vitamin A. So let's start by the metabolism. How does we acquire our needs from vitamin A? So the vitamin A is present all over in different parts of the body, but it is the highest in the liver and the RP. These are the main parts or the main organs for storage of vitamin A. Also, vitamin A is present in the conjunctiva, in the cornea. It is important for the healthiness of the cornea and the conjunctiva, but it is present in much more amount in the liver and the RP. So the liver will be the main site for the storage, and it will be transferred from the liver to the RP. So in the liver, it will be transferred as a specific form called old trans retinyl esters. Old trans, it is just a name for the position of certain molecules attached to the vitamin A molecule itself. So it is just a chemical formulation. This is called all trans. Retinol esters, which is a form of vitamin A formed by stratification. This is mainly or basically it is retinol binding or binded with fatty acids. Then as it's present in this form in the liver, it will be now formed or transferred from the liver to the RPE through the plasma. In the plasma, it will be bound to a plasma protein. This is called serum retinoid binding protein, and this will in turn will bound to a larger prealbumin molecule. So this is very important, that what will hold what will transfer the vitamin A from the liver to the RPE, it will be the serum retinoid binding protein. Let's see here. Well, here we have in the intestinal cells. We can in the diet we take the beta carotene, and this is the inactive form, and in the intestinal cell it will be transferred. It will be transformed into retinal and retinol, and then retinol esters. Or if we take the retinol immediately from the diet, so it will be absorbed as a retinol, and it will be formed by or will be stratified into retinol esters. Then it will be transferred into through the lymphatics to the liver, where it will be stored in the liver as all trans retinol. So all trans retinol, or the retinol, stored as retinol esters mainly in the liver and the adipose tissue. So it will be formed or will be stored as all trans retinol esters. Then from this location will be the liver will be the main source, and then it will be transferred to different tissues, like for example, as we said, in the epithelium and in the conjunctiva, it will be transferred into smaller amounts in the target tissues where it will produce their action, but the main bulk will be transferred into the retinal pigment epithelium. So in the retinal pigment epithelium, it will be transferred by the retinal binding protein, where it will be entering through the retina, and it will undergo will be a part of the visual cycle, as we will talk now. So where does the vitamin A in the RP comes from? First of all, we said from the plasma, as the basal membrane of the RPE contain receptor for the serum retinoid binding protein. So let's say, for example, that this is the RPE cells, and here is okay, let me make it a bit higher. So this is the RPE cells, and this is the blood in the choriocapillaris. So the basal part here of the RPE cells have some receptors, these receptors for the serum retinoid binding protein. So if there is a serum retinoid binding protein in the circulation, this will lead to entering of the RPE to the cell. The other thing is that these photoreceptors are interdigitated with the outer segment of the photoreceptors, so that during the process of bleaching, as we'll say now, this will lead to breaking down of the photoreceptor outer discs, so that this will lead to liberation of the vitamin A, which is present here, into the cells. Also, it will be liberated inside the RPE. This is during the phagocytosis of the outer segment disc shed during the renewal and shedding, and also liberated into the receptor outer segment by bleaching the visual pigment, and transferred to the retinal pigment epithelium by interstitial retinoid binding protein. You see that there will be a potential space between the outer segment photoreceptors and the retinal cells themselves, so that if there is any liberated vitamin A in this interstitial space, it will be transferred into the retinal pigment epithelial cells and will be using an interstitial retinoid binding protein. It is another protein that will bind this free vitamin A, and the apical part of the RPE cells have receptors for this interstitial retinoid binding protein. So from below, it will come from the serum retinoid binding protein. From above, it will come from the interstitial retinoid binding protein. And from the shedding of the photoreceptor outer segment, as they are phagocytosed inside, so this outer segment will also contain vitamin A. So it's a bit of clinical information. Why do or what will be the problem if we don't have excess or enough amounts of vitamin A? This will result in a disease or a group of diseases called nyctalopia or night blindness. And this occurs due to severe deficiency of vitamin A, so that this process of rhodopsin formation and the visual cycle processing will be interrupted due to lack of the vitamin A. So that this will lead to affection of night vision, as the light sensitive molecule, which is vitamin A or light sensitive molecule of rhodopsin, is consisting mainly of a form of vitamin A, as we will see later. So fortunately enough, this is not very common, because it needs a lot of or a severe deficiency of vitamin A for people to develop nyctalopia, and will need to the stores of vitamin A to be depleted for several months until the clinical picture appear. And also, fortunately enough, this can be reversed in even by single injection of vitamin A. So what are the manifestations? It will be present with what's called the B2 spots, corneal ulceration, and xerosis and corneal melting, and what's called keratomalacia. So this is just a side information as we talk about the vitamin A deficiency. So let's now talk about what is the most important part in the lecture, which is the visual cycle. How does we generate the visual impulse in the retina? This can be done with the aid of what's called visual pigments. So what are the visual pigments? These are pigments, or these are molecules or substances that have the ability to absorb the light. First one is the rhodopsin. And the rhodopsin, it is the light-sensitive molecule present in the rods. And it will decay, or it will decompose after exposure to light. So it is composed of two main parts. The first one is a protein called the scotopsin, and a form of vitamin A, it's called retinal. Both of them will form the light-sensitive molecule that is called rhodopsin. It has a molecular weight of 40,000 and fat-soluble. It is sensitive to heat and the chemical agents, and the scotopsin itself, it is formed of 348 amino acids that cross the disc 7 times. These 7 times have been asked before in the MCQ. So let's see. Here is the rhodopsin. It is the light-sensitive molecule present in the rods, not the cones. And it is a transmembrane protein, as you can see, that is formed by like opsin. We can call it opsin or scotopsin, and the retinal, which is a form of vitamin A. Retinal, this is the black dots here, the small black dots, this is the retinal. And the opsin or the scotopsin is a transmembrane protein that cross the protein 7 times, or cross the membrane 7 times. Again, here is the membrane of the disc, of the outer segment of rods, and here is the retinal, the form of vitamin A, and here is the scotopsin, which is crossing the disc 7 times, or crossing the outer cell membrane of the disc 7 times. The form of retinal, it is called 11-cis retinal. So 11-cis retinal, it is the form of vitamin A specifically, or the specific molecular configuration of the retinal present in the vitamin A or present in the rhodopsin. It's called 11-cis retinal. Since that this is a light-sensitive molecule, so that it will have it will absorb the light. And every visual pigment will have what's called the spectral sensitivity. So spectral sensitivity meaning that it has a peak sensitivity for a specific wavelength. And in case of the rhodopsin, it has a peak sensitivity around 500 nanometer, which is more toward the yellow wavelength. So it will absorb or have high sensitivity to the yellow wavelength, and once it is absorbing this yellow light, it will transmit violet and red, so that it appears purple, and that's why it will be called a visual purple. Sometimes the visual pigment of the rods or the rhodopsin, it is called the visual purple. And since that the rhodopsin is very sensitive to the light, the yellow wavelength, we can use this for different. We can use the yellow color in illustration for taxi sign or for traffic signs, as the yellow color will be very clear during the dim illumination or during the night, because the rods have a high spectral sensitivity to the yellow light. The cone pigments are not very intensively studied like the rods, so we don't know what are the exact molecular structure of the cone pigments, and we don't know what are the configuration of the cone pigments, but we know that we have three types of cone pigments that are different from rhodopsin, because they have different spectral sensitivity, so they respond to different wavelengths like the red and the green and the blue. And the opsin itself, it is almost identical. So it will be almost identical except for few amino acids that make the difference in the spectral absorbance. So they have opsin as well, or they have, but this is in such case it's not a scotopsin, as it is not used for night vision, but it is an opsin that have different configuration in slight amount of amino acids that will make the difference in the spectral sensitivity. 11-cis-retinal, or the form of vitamin A, will be the same, so the cone pigments will be different from the rhodopsin by just these few amino acid sequences. And they will have different peak absorption for blue and red and green. And we have to know from now that the blue sensitive cones are the least prevalent. So the least amount of cones in the retina will be the blue cones, not the red and not the green. So as we can see here, this is a curve showing the spectral sensitivity of the short wavelength and the medium wavelength and long wavelength. So short starting from the blue by about 450 microns, while the green showing spectral sensitivity to about 550, and the long wavelength that's about 600 nanometer. So for each one of them, or when the light hit let's say for example for the red, if the light hit the retina, which has a red or have a spectral have a wavelength of 600, this will cause stimulation of the cones by different amounts according to the spectral sensitivity. So for example, it will stimulate the red cones with about 100%, the green cones by about 70%, the blue cones by about 20%, as this is the way that our cones are stimulated. Also, for example, let's say for example that there is a light which has a peak wavelength of 500, or the wavelength of the light is 500 nanometer. So it will stimulate the red light or stimulate the red cones like 30%, the blue cones by about 40%, the green cones about 50%. So the resultant light will be like yellow. So how these cones work. So the light falling on the retina is absorbed by the photosensitive pigments and initiate photochemical changes that will initiate the electrical changes. This is what's called the visual cycle. The visual cycle is that we have visual pigments present in the photoreceptors. When they are exposed to light, this will result in photochemical changes of these visual pigments, and this photochemical changes will lead to electrical changes, or will lead to formation of an electrical current. And this process is called the phototransduction. The change of the photochemical reaction to electrical charges called the phototransduction. So in order for these photochemical changes in the outer segment, this will undergo two processes. The first one is called the bleaching. Bleaching is the decomposition of the visual pigment under the effect of light, so that we can see in the light. And then these visual pigments, if there is no light or in the dark, these visual pigments will regenerate in order to be able to bleach again under the effect of light. So continuous process between bleaching and regeneration, this is what's called the visual cycle. Again, visual cycle consists of two main phases. One occur under exposure to light, it is called bleaching, in which there is a photochemical changes of the visual pigments that will result in formation of electrical changes or electrical charges. Then under the dark, there will be regeneration of the photopigments. Okay, let's now start by the rhodopsin visual cycle. Both rods and the cones have chemicals. When they exposed to light, they will decompose, leading to excitation of the nerve fiber from the eye. In the rods, the light sensitive is rhodopsin, as we said. In cones, we have what's called the cone or color pigments. We can call them color pigments, or we can call them the erythrolabe, which is the red sensitive, chlorolabe, which is the green sensitive, and cyanolabe, which is the blue sensitive. So the visual pigments, these are the visual pigments, and we will talk mainly about the rhodopsin. This is one, as this is the one which is the studied intensively, and the one which was present in the books and asked in the exams. So the rhodopsin or the visual purple is present in the outer segment of the retina. We said that we have discs in the outer segment. These discs have a transmembrane protein called the rhodopsin molecule, which consists of opsin or scotopsin and consists of a form of vitamin A called 11-cis retinal. The 11-cis retinal, it is bind to scotopsin to form this molecule of rhodopsin. So what happens when there is exposure to light? When there is exposure to light, there will be what's called rhodopsin bleaching. Bleaching, as we said, it's called activation, or this is make the rhodopsin an activated rhodopsin. So how can we make the rhodopsin an activated rhodopsin? This is a chemical process, as we said. So this chemical process is called photoisomerization. In this photoisomerization, we change the form of vitamin A to another form. So we change the 11-cis retinal plus opsin, which is the rhodopsin molecule, into all trans retinal and free opsin. Don't confuse this with all trans retinol present in the liver. So it is, again, it is the process of formation or changing the rhodopsin molecule, which is basically a combination between 11-cis retinal and opsin, into all trans retinal plus free opsins. So what happens? When the light falls on the rhodopsin molecule, this will lead to it will decompose in a fraction of a second, very rapidly, to form an intermediate molecule that's called a bathrhodopsin. Then the bathrhodopsin will change into lumirhodopsin. Then the lumirhodopsin will change into metarhodopsin 1. And then the metarhodopsin 1 will change into metarhodopsin 2, which is called the activated rhodopsin. So again, the rhodopsin under the exposure of light will decompose so that the opsin molecule will leave the vitamin A, or the rhodopsin will change to the bathrhodopsin, then the lumirhodopsin, then the metarhodopsin 1, then the metarhodopsin 2, and the metarhodopsin 2 is called the activated rhodopsin. And this is the molecule which will induce or stimulate the electrical changes in the rods that will be transmitted to the bipolar cells and to the ganglion cells, and will be transmitted as a visual signal to the CNS. So this is the molecule which will be responsible for formation of another sequence of events we will talk about now, that will be responsible for electrical impulses. that will reach the brain. So what happens next? Well, then if we have this will be the only process that will occur. So we eventually at one time we will have all our rhodopsin depleted, so it will all finish. So we need to regenerate this molecule of rhodopsin. So let's see what happens now. In the dark state, as we stay in the dark, this is the molecule of 11-cis-retinal, and this is called the inactive rhodopsin form. Under exposure of the light, this will form the bathrhodopsin and the lumirhodopsin and the metarhodopsin 1, which is again inactive. That will form the metarhodopsin 2. The metarhodopsin 2, this form is the active form, which will cause what's called the activation of protein G, as we will see now, and will be responsible for the electrical impulse formation. And then this metarhodopsin 2 will undergo a sequence of chemical events that will lead to formation or reformation or regeneration of the rhodopsin. So as you can see here, the rhodopsin is decomposed, so it will lead to formation of the form of vitamin A, which is called the meta 1 and the meta 2, and the opsin. Don't forget about the opsin, that the opsin is now free in the disk. So where is the vitamin A and what is the vitamin A role here in rhodopsin formation? You can see that the metarhodopsin 2, it is called all trans retinal. This is the active form. So by formation or by changing the 11-cis-retinal to all trans retinal, we have now an active form of rhodopsin that will lead to initiation of the visual impulse. We need now to return this all trans retinal into 11-cis-retinal. So let's see how does this occur. The all trans retinal can be converted to 11-cis-retinal by an alternate route. This is to convert the all trans retinal to all trans retinol, which is another form of vitamin A. Then all trans retinol is changed to 11-cis-retinol under the influence of isomerase. Finally, the 11-cis-retinol is converted to 11-cis-retinal. So the activated rhodopsin or the metarhodopsin 2, which is the all trans retinal, can be either transformed to 11-cis-retinal through direct mechanism, which is through the isomerase, or it can transform it into all trans retinol, which is a form of vitamin A. This occurs in the RPE, and then within the RPE cells, it will, under the effect of the isomerase enzyme, it will transform this all trans retinol into 11-cis-retinol, and then 11-cis-retinol will be transformed into 11-cis-retinal. So now we can form our visual pigment again. So you can see here, this is the retinal pigment epithelium, and the all trans retinol here is transformed into all trans retinal, and then finally to 11-cis-retinal. So we can change then the 11-cis-retinal will bind with the opsin in order to form the rhodopsin again. So again, the regeneration of the rhodopsin, meaning that we can convert the all trans retinal into 11-cis-retinal. And this requires energy and the enzyme retinal isomerase, and the 11-cis-retinal will automatically combine with the scotopsin to reform the rhodopsin, which is occurring in the darkness. So here you can see that the rhodopsin molecule, which is consist of 11-cis-retinal and scotopsin, under the effect of light, this will lead to its decomposition. So its decomposition will lead to scotopsin, or the opsin will be free, and the vitamin A will undergo a series of changes. It will form the bathrhodopsin, then the lumirhodopsin, then the metarhodopsin 1, then the metarhodopsin 2, which will be the activated rhodopsin. This will initiate the electrical impulse, and then it will convert, or it will form the all trans retinal. By the effect of isomerase, it will form the 11-cis-retinal. The 11-cis-retinal will bind with the scotopsin again, and will form the rhodopsin. This process occurs in the dark. Or the all-trans retinal will be transformed to all-trans retinol, which will be again under the effect of the isomerase will form the 11-cis retinol to convert to 11-cis retinal and will bind again with the scotopsin to form the rhodopsin. Don't worry, you will memorize it. We have plenty of examples. So in the rod outer segments, this is where occur the process of bleaching and regeneration. So this is the rhodopsin molecule consisting of opsin bind with 11-cis retinal. What happens when the light strikes the retina? Then this will lead to decomposition of this molecule, this decomposition of the rhodopsin into free opsin and other forms of vitamin A, bathrhodopsin, lumirhodopsin, metarhodopsin 1, metarhodopsin 2, that this is called the activated rhodopsin. Then the activated rhodopsin will be transformed into all trans retinal, and the all trans retinal will be changed to the 11-cis retinol, 11-cis retinal that will bind with the opsin again to reform the rhodopsin molecule, and this occurs in the dark. Again, in the outer segment, here is the 11-cis retinal, and here is the all trans retinal. Under the effect of light, the photon of light will cause formation of all trans retinal, which will be the active form. Then the all trans retinal will be converted to 11-cis retinal, which will bind with the opsin again to form the molecule of rhodopsin, which will occur in the dark. This illustration will show which process occurs in which place. So in the photoreceptor, under the effect of light, the rhodopsin molecule will break into the 11-cis retinal and opsins. So the rhodopsin molecule will convert to the metarhodopsin 2, which will be responsible for initiation of the neural signal and vision, which will pass through the bipolar cells and bipolar and ganglion cells and the rest of the visual pathway. This occurs in the light. But during the dark, the regeneration of the rhodopsin molecule occurs. So this occurs by formation either that all trans retinal will transform into 11-cis retinal immediately through the isomerase enzyme, like this, or another pathway that it will be transferred into the retinal pigment epithelium. So this the all trans retinal will be transformed into all trans retinol. This all trans retinol coming from either the pigment epithelium, coming from the outer segment disc, or coming from the capillary. You remember the serum retinoid binding protein. This will be binding to all trans retinol. They will be stratified again into the retinal pigment epithelium into 11-cis retinol. Then it will be forming 11-cis retinal that will return back into the outer segment as 11-cis retinal that will bind with opsin to form the rhodopsin molecule. Is everything clear up till now? Hello? Okay. So we now know what is the visual cycle. So how does the rhodopsin decompose and how does it regenerate? So now we need to know how does the electrical impulse generate. This is in this case we need to understand that there is what's called the rod receptor potential. Potential meaning that there is an electrical charges or electrical current across the membrane of the rods. Normally there is the rod receptor potential in the dark, it is about minus 40 millivolt. So in this condition, it is a slightly depolarized or even hypopolarized. This means that the membrane is slightly negative. This occurs because the outer segment of the rod is very permeable to sodium ions. And in the dark, there is inward current that is called the dark current that is occurred due to the sodium influx into the outer segment of the photoreceptors or outer segment of the rods. Let's see what does this happen or what does it mean. Again, in the dark, there will be a depolarized membrane or depolarized rods. This occurs due to the sodium ion is entering inside the disc or entering inside the receptor, leading to that there is a slight negativity of minus 40 millivolt of the membrane of the photoreceptor. But when there is a light exposure or when there is the rhodopsin decompose forming the metarhodopsin 2, this will lead to hyperpolarization of the rods, leading to by decreasing the sodium permeability of the outer segment. And the greater the amount of the light, the greater the amount of electronegativity, meaning that the membrane was slightly negative, about minus 40 millivolt, and this what will cause depolarization and in the resting state. But when there is exposure to light, this will lead to increasing the negativity by decreasing the sodium permeability of the outer segment, leading to increasing the negativity to minus 60 or minus 70, which will lead to hyperpolarization. And hyperpolarization here means activation and processing of the electrical charges. So let's see here what happens in the dark and what happens in the light. In the dark, there is mild negativity of the rod membrane. As you can see here, this is the rod inner segment, and this is the 0. So this is a slightly negative, about minus 40 millivolt. This occurs because, as in the dark, there is a gate called the cyclic GMP, which is open and allowing the sodium to enter in the cell. So since the sodium, which is positively charged, enter inside the cell or enter inside the rods, this will lead to that the cell is mildly negative or have mild negative charge or slightly depolarized. But what happens in exposure to light, this will lead to formation of the metarhodopsin 2. And the metarhodopsin 2 will cause closure of this cyclic GMP gates. So when the sodium is trying to enter inside the cell, it will be returning back. So this will lead to that there is the inside of the cell will be much more negative as the sodium is not able to enter, leading to increasing the negativity, leading to initiation of the electrical impulse by hyperpolarization. So what is the sequence of events that occur? This is the process of phototransduction. The instant light or the photon of light will activate the electron, leading to structural changes in the rhodopsin, leading to changes in the photopigments, leading to activation of the protein G called the transducin. The transducin will stimulate an enzyme called the phosphodiesterase. This phosphodiesterase will cause destruction of the cyclic GMP. So the cyclic GMP phosphodiesterase will destroy the cyclic GMP. So what is the cyclic GMP? The cyclic GMP is a gate responsible for keeping the sodium channel open. So when the light activates the rhodopsin or the metarhodopsin 2, this will activate the G protein or the transducin. The transducin activates the phosphodiesterase, destroying the cyclic GMP, so that there will be decrease in the intracellular cyclic GMP, leading to closure of the sodium channel and hyperpolarization. This hyperpolarization will cause decrease the release of the synaptic neurotransmitter or glutamate, leading to a response in the bipolar and other cells like horizontal or amacrine cells. So if there is hyperpolarization, this will lead to decrease the glutamate. If there is depolarization, there will be excess glutamate. So the glutamate neurotransmitter, it will be the neurotransmitter which will be responsible for initiation of the electrical impulse. The rhodopsin kinase enzyme then will deactivate the activated rhodopsin. This is the negative feedback mechanism. So this can be initiated by the rhodopsin kinase enzyme. Will deactivate the activated rhodopsin. Then the cyclic GMP will be regenerated, leading to reopening of the sodium channels. So this is the sequence that we were discussing. Let's see it. So what happens in the dark? In the dark, the cyclic GMP will be gates will be open, so there is a sodium influx inside the outer segment of the photoreceptor, leading to formation of what's called the dark current, making the membrane potential is slightly negative, about -30 to -40, leading to depolarization, and this depolarization will result in glutamate release at the neurosynaptic junction between the photoreceptors and the bipolar. And in this case, this will lead to inhibition of the visual impulse. In case of activation of metrorhodopsin 2, this will lead to destruction of the cyclic GMP. So the sodium or the gates will be closed, so the sodium will not be able to enter inside the cell. This will result in increasing the negativity, so that it will be about -40 to -70, so that there is no sodium inflow inside the cells. This will lead to hyperpolarization. In case of hyperpolarization, this will lead to decrease or stoppage of the glutamate release. And since that the glutamate is stopped, this will lead to excitation of the bipolar cell as the impulse will continue from the photoreceptor to the bipolar cell. So here is the sequence again. Again, in the dark, there will be a passive influx of the sodium, decreasing membrane potential, and this will lead to a secretion of excess glutamate at the neurosynaptic junction. But in case of light, the light will cause activation of rhodopsin that will activate the transducin or protein G, that will activate the phosphodiesterase enzyme, that will cause destruction of the cyclic GMP, leading to closure of the sodium channels. And then in such condition, this will lead to hyperpolarization, and the hyperpolarization will cause the glutamate release to stop and will be leading to transmission of visual impulse from the photoreceptor to the bipolar cells. We have said this. This is the same thing. And this is the same thing, the whole process again. In the dark, the rhodopsin will decompose to opsin and 11-cis-retinal that will form the all-trans-retinal, which is the active molecule that will stimulate the transducin, stimulated phosphodiesterase enzyme, leading to closure of the cyclic GMP gates. So the sodium will be closed, so it will not be able to enter inside the cells, leading to hyperpolarization and leading to stoppage of glutamate, so the impulse will be transmitted to the bipolar cells. We will see many examples so that you can understand the whole process again and again and again. So in the dark and in the light, in the dark and in the light. So do you have any question regarding this, the visual cycle? Is everything clear? Okay, so let me ask you, which one of them will cause or will present in the dark, depolarization or hyperpolarization? So what happens on exposure to light? It is depolarization or hyperpolarization. Exactly. So remember that in the light there is hyperpolarization, and in the dark there is a depolarization. And let me tell you a surprise now, that this is only at the level of photoreceptors, because we will reverse this as we will talk later on. So remember this slide when we were talking about the transmission of the visual impulse. So the vision is starting from the inner to the outer, but or the transmission of light is from the inner to outer, but the transmission of the visual processing will be from the outer to inner, so it will be in the retrograde manner. So we are here. We discussed what happens here now, what happens in at the level of the outer segment photoreceptor in the term of visual cycle, what happens in the term of the sodium gates, and this now we are here now. We are at the neurosynaptic junction between the rhodospherule and the end of the bipolar cell. So glutamate will be released in case of the dark current, so there will be excess glutamate, which will prevent the transmission of the electrical impulse. But in case of under the effect of the light, there will be very few or no glutamate, which will facilitate the transmission of the electrical impulse. So again, this is the direction of light, but this will be the direction of the visual processing. So as we said, in case of the dark, there will be a depolarization, so that the resting membrane potential will be about minus 40 millivolt, but under the effect of the light, there will be a hyperpolarization reaching to minus 60 millivolt. So this will occur in the photoreceptors. So what happens next? First of all, the electrical impulse will pass through the retina in the form of electrical current, because there is no true action potential except in the ganglion cells. So the ganglion cells are the only cells that will have a full action potential. But in case of the other retinal cells, they have only electrical current, either depolarization or hypopolarization or hyperpolarization, but not a full electrical action potential. And this electrical current will be graded according to the intensity. So the higher the intensity of the light, the much more the response and the much more the hyperpolarization occur at the photoreceptors. So this can be considered like a graded response. Now we come to the bipolar cells. So what is the pattern of receiving the stimulus in the bipolar cells? When the bipolar cells receive the visual impulse from the electrical impulse from the photoreceptor, they will follow a configuration called the center surround organization. So what is the center surround organization? This means that when the bipolar cells will have a configuration or a horizontal configuration of difference sensitivity between the center and the periphery. So they form what is called a receptive field. The bipolar cells will form what's called the receptive field, so that when this receptive field have a center and have a periphery, sometimes the center can be excitatory, meaning that stimulation of the center will cause stimulation of the bipolar cells. And it will have an off surround or an inhibitory surround. The inhibitory surround meaning that stimulation of this periphery will cause inhibition of this bipolar cells. In such a condition, if there is the center is excitatory and the periphery is inhibitory, it is called on center of surround, or we can call it on cell. So we call the cell by the name of its center, or we can call it on center of surround. What is the on center of surround? This is a receptive field or a group of bipolar cells. When they have the central part is excitatory and the peripheral part is inhibitory. So stimulation of the central part will cause stimulation of the bipolar cells. Stimulation of the peripheral part will cause inhibition of the bipolar cells. Conversely, there is a converse type. It's called the off-center on surround. In the off-center on surround, it is a receptive field that has an inhibitory center and an excitatory periphery, and this is called off cells. The off cells, when stimulated in the center, This will lead to inhibition of the bipolar or inhibition of the electrical signal transmission. If they are stimulated in the periphery, this will cause facilitation of the electrical impulse. And we also with the function of each receptive field is in case of physiology is the function of the center. So this is called on cell and this is off cell. So visual impulse reaching the receptive field of the on cell bipolar will cause a facilitation of this or will cause a facilitation of the transmission. Visual impulses related to the or received by an off bipolar, this will lead to stoppage of the electrical impulse. This is what's meant by the receptive field. So when we have the different cells, these different cells will have a center surround configuration, meaning that the light is entering or will striking the center of this photoreceptor or this cone will lead to a center or an on response. While the light striking the periphery of this or the surround of this receptive field, this is called giving what's called off response. So this is what's called the center surround configuration or the receptive field. I hope that is clear because we will need this concept right now. So in bipolar cells, the reverse occur, the pattern of stimulation will be the reverse like the photoreceptors. In case of bipolar cells, the depolarization will mean that there is a stimulation, while the hyperpolarization will mean that there is an inhibition. So let's say, for example, that if there is a light, in case of light, there will lead to hyperpolarization at the level of photoreceptors. So this will result in decreased glutamate. So what happens when this photoreceptor is attached to an on center of surround bipolar? Then this will stimulate the on center. And this stimulation will be by depolarization. So depolarization here occurs in the on center of surround bipolar, while if the light is striking an off center on surround, this will lead to hyperpolarization. So hyperpolarization meaning that there is an inhibition of the inhibitory part of the center, which is off. So inhibition of the inhibition will lead to facilitation of the impulse. Again, here we have the light striking the photoreceptors, causing hyperpolarization. In order for this light to be transmitted through the visual pathway, this will pass through either on cells or off cells. The on cells or on center, if the light is striking the on center, this will lead to depolarization, leading to facilitation. Or if the light is striking the off center, this will lead to hyperpolarization of the center. So in hyperpolarization of the center, this will lead to inhibition of the inhibitory cells. So this will result in facilitation of the electrical signal transmission. So bipolar cells are the first order neuron in the visual pathway. The on bipolar, under the effect of light, this will result in decrease the release of glutamate, causing stimulation of the bipolar cells of the on bipolar by depolarization, causing stimulation of the ganglion cells. In the off bipolar, the light will decrease the release of glutamate from the end of photoreceptor, leading to inhibition of the bipolar, leading to hyperpolarization. This will lead to, again, inhibition of the bipolar cells, which is the off. So inhibition of the inhibition will cause, by hyperpolarization, will cause facilitation. So this is stimulation, not inhibition. So the on bipolar cells and the off bipolar cells will both work together under the effect of light in order to facilitate the electrical signal transmission. So to summarize this, under case of light, there is hyperpolarization of the photoreceptors, leading to decrease the release of glutamate, leading to depolarization of the on cell, which has a central on and surround off, or central. The central on will be depolarized, causing facilitation of the electrical signal. In case of the off center and on surround, this will do again the hyperpolarization or stimulation of the photoreceptor will release less glutamate, so the glutamate will be less, and this will lead to stimulation of the center. So the center here will be hyperpolarized and will be inhibited. So there will be inhibition of the inhibitory bipolar cell, leading to facilitation of the electrical signal as to reach the ganglion cells. The reverse will occur in the dark. In the dark, there is a depolarization of the photoreceptor. So this will lead to release of excess glutamate, causing hyperpolarization of the center of the on bipolar, leading to stoppage of the electrical impulse. Or if there is depolarization causing again excess glutamate, leading to stimulation of the off center of the bipolar, causing depolarization, so it will lead to again to stoppage of electrical impulse. We are now talking about the light. Dr. Suham is asking about the inhibition of ganglion cells. We are talking about the off bipolar, and the off bipolar, when stimulated by the light here will cause inhibition of the inhibitory cells. So it will cause facilitation of the electrical impulse by hyperpolarization of the inhibitory cells. So it we prevent the inhibitory cells from action. So that will result in stimulation of the ganglion cells, not inhibition. But inhibition of the ganglion cells doesn't occur in the light, it occurs in the dark. Okay, so now we come to the, after passing the impulses through the bipolar cells, or we reach the bipolar, now we come to the horizontal cells. The striking rule of the horizontal cells that's called what's called lateral inhibition. Lateral inhibition is used for enhancing the visual contrast. So as the horizontal cells are spreading laterally, they connect the rods and the cones so that they can modulate the amount of electrical charges. Unlike the bipolar cells and the photoreceptors, they don't have excitatory and inhibitory. They are always inhibitory. This is very important. And they prevent the lateral spread of light excitation on the retina, leading to formation of a contrast or enhancing the contrast of the image. So let's say, for example, that the horizontal cells are acting like this knob, or this graded knob. The graded knob, or if we have a graded knob, we can increase the intensity and decrease the intensity of electrical impulse in a graded manner. And this will lead to decrease the contrast between the edges of the image. But let's say, for example, that we have a black and white image, and the junction between each of them, we block, or we switch the button off between the edge of the black and white. This will lead to enhancing the contrast, as the black appears very black and the white appears very white. And this will be the result, or the function of the horizontal cells. So here we have the photoreceptors. The photoreceptors will cause excitation of the bipolar cells, and here we have the horizontal cells are acting horizontally. By placing horizontally, they will do inhibition at certain bipolar cells, so they will prevent the lateral spread of impulse to both sides. So for example, if we have the excitation, and for example, if we increase the excitation, this will be with no light after putting the light on, and if there is a light off, and if there is no horizontal cells, there will be a minimal difference between the on and off. As you can see, this is the on and this is the off. There is no good or no great difference between both of them. But in case of the horizontal cells, this will lead to switch off the bipolar cells, so that there will be a greater difference between the on response and off response. I made this illustration so that you can see, understand what is the idea about lateral inhibition. If there is no horizontal cells, you can see the image like this, or you can see all images in a gradient manner. But since that we have horizontal cells, you can see sharp and clear edges of each object. So if you see an object, you will not find its edge hazy, because the bipolar cells or the horizontal cells here cause the lateral inhibition of the visual impulse at the edges of this object. What is the unclear point, Dr. Amira? Okay. Previously, if I didn't talk about the on-center and off-surround, you will suppose that stimulation of one photoreceptors will cause stimulation to one bipolar cells, causing stimulation to one ganglion cell. And the inhibition of the photoreceptor will cause inhibition of the bipolar cell and inhibition of the ganglion cell. Is that right? So you would think so. But actually, this is not the case in case of the retina. The retina doesn't work like this. But retina work as groups. As we said, the process of convergence. So you remember when we were talking about the convergence? In the convergence, meaning that there is a different or large number of the photoreceptors will converge to a single bipolar cell. So this bipolar cell will process information from different number of photoreceptors, and sometimes different or large number of the photoreceptors will convert, like for example, like 100 photoreceptors will converge to about 3 bipolar cells. So here's one bipolar, and here's one bipolar, and here's one bipolar. This 100 photoreceptors will follow what's called center on and or center surround configuration, meaning that stimulation of the center of this photoreceptor will cause stimulation of one bipolar in the center, and this will result in inhibition of the photoreceptors in the periphery, resulting in inhibition of the bipolars in the periphery. This will result in the image will be showing more clear and more sharp edges, as there will be a clear contrast between the center and the periphery. And this will be helped by the horizontal cells. These horizontal cells will connect the bipolar cells together, so they will modulate, they switch some bipolar on and switch some bipolar off, so that they will improve the contrast of the image. You are asking when to be on. This is there is no a sharp rule. So this depends on what is the stimulus you are seeing. Is it large or it is small? Is it of high intensity or low intensity? And how it is represented on the retina. So sometimes the single photoreceptor can be on, and sometimes it can be off. This is this is a thing that the retina can arrange in order to improve the image. So we don't have a specific number of on cells and off cells. We don't have a specific anatomical configuration for on cells and off cells. This is a changing process according to the electrical charges all the time. So is this still vague, or it's clear, or what's your confusing point? So we just okay, thank you. We just need to know what is the mechanism of the on cells and off cells. That's what we concern us in the physiology. not or to understand that there is an on cells and off cells, and there will be the on cells in case of, or the depolarization will cause stimulation in bipolar, while the hyperpolarization causes inhibition. Then after the horizontal cells, we have another cells called amacrine cells. The amacrine cells are not very extensively studied, and they found that there is more than 30 types of amacrine cells that are again involved in modulation of the response between the rods, in the modulation of the visual transmission. Some of them can be a direct pathway from rods to bipolar to amacrine to horizontal, so that they act as a station in between the rods and the ganglion cells through which the light impulse will pass. Others will be responded to the onset of the visual stimulus. So with the initiation of the visual impulse, the amacrine cells will respond. Some respond to the movement of the signals across the retina. And amacrine cells are a type of interneuron that aids in the beginning of the visual signal analysis and what's called the temporal summation. We will talk about it later. So the amacrine cells are a heterogeneous group of cells that have different functions, all to modulate the visual impulse, either in the start or through the movement, or can be a station in between. These are called the amacrine cells. So to compare between the horizontal cells are amacrine cells. The horizontal cells are laterally connected the rods and the cones and the bipolar, while the amacrine cells laterally connect the bipolar cells and the ganglion cells, which are more than 30 types. The horizontal cells will be responsible for inhibition of lateral excitation of the electrical impulse, what's called lateral inhibition, leading to increasing the contrast and increasing the visual accuracy. While the amacrine cells have different functions, like being stimulated in the initial part of the stimulus, or stimulated during the movement of the stimulus, or help in the temporal summation. Then the final part, or the second order neuron in the visual pathway, will be the ganglion cells. So the ganglion cells will be about 1.6 million in numbers, and they are, as we said, it will be the only retinal cells that will respond by a full regenerative action potentials, as all other cells will be responding by an electronic current. So once they are stimulated, they will respond by full electrical potential. We have three types of ganglion cells. So we have the W cells, we have the X cells, we have the Y cells. The W cell will be forming about 40%. The X cells will be the most numerous. The W cells will be 40% and will be a small cells, while the X cells are the most numerous, making about 55%, and the medium cells. The Y cells are the least numerous and the largest. So it's making about 5% only, but it is the largest cells. They follow the rule of receptive field. So the ganglion cells doesn't respond on cellular level, but respond on a field level, or a group or a collection. Like, let's say, for example, that we are to better understand the idea of receptive field and on and off. When we are, let's say, for example, that you are looking to the retina from anterior to posterior. So here is the retina. Here is the optic nerve and the arcades, and here is the fovea. So there will be here a ganglion cell, and the ganglion cell, and the ganglion cell, ganglion, and different or numerous ganglion cells. When there is an impulse stimulating these ganglion cells, or stimulating the rods, and then transmitted to the bipolar, transmitted to the ganglion cells, all this act as a single unit. All these ganglion cells act as a single unit. This is called a receptive field. This receptive field, the peripheral part of this receptive field, can be negative, or what is called off. So that stimulation of the periphery of this ganglion cells, of this group of ganglion cells, will cause inhibition of the impulse. And the center is called on, or positive. So the on center of surround, by stimulation of the center of it, this will cause stimulation of the or transmission of the visual impulse. So this receptive field is present in the bipolar, present in the ganglion cells. In case of W cells, they have larger receptive field, while in the X cells, they have smaller receptive field, and in Y cells, they have, again, larger receptive field. The W cells receives their input from rods, so that since the rods are more numerous and more dispersed through the retina, this will lead to that these W cells will be responding more to the directional movement in the visual field. So if there is any movement or any change in the movement in the visual fields around you, this will be received or perceived by the W cells. While the X cells will be sensitive to the directional movement in the visual field. Sorry, this is just I didn't write it well. Hold on, please. Okay, sorry for interruption. That I just updated this lecture today, so this was not. I didn't update this. Okay, no problem. So again, back to the types of the ganglion cells, we have the X cells will be responsible for color vision. We will talk about the color vision in a separate lecture tomorrow, inshallah. And we will say that it will be received or perceived by the X cells of the ganglion cells. The Y cells will be responsible for rapid changes in the visual image. Like, for example, if there is rapid change in the movement, like the object has suddenly become slower or suddenly become more rapid, then this will be perceived by the Y cells. Or even if the object become more intense or the light intensity become more or even less, this will be perceived by the Y cells, and it will receive the input from rods and cones. So as we said, the rods, since they are more numerous, they will be able to perceive the movement in the space better. The cones, since they have red and blue and yellow cones, this will be perceiving the color vision better. The rapid change in the visual image will be perceived by both rods and cones. You may find in some other books there are different or another distribution or another classification of the ganglion cells into M cells, P cells, and K cells. So I prefer to let you know also about this classification, which is more anatomical rather than functional. The M cells is less common, and the P cells are the most numerous, and the K cells are only involved in the blue color. The M cells are corresponding like the W cells, so they are having large receptive field corresponding to about 200 of bipolar cells. And this is responsible for gross analysis of the visual image and location of the object in the visual field. So, this is like the W cells. The P cells are more than numerous and have a smaller receptive field like 1 to 1 or few bipolar cells and responsible for fine detailed vision for the shape and texture. So for some books, they say that the M cells will projected to the magnocellular and magnocellular layers in the lateral geniculate body, and the B cells will project to the parvocellular layers in the lateral geniculate body, and hence they are named M and the P cells. And the magnocellular layer will be responsible for the cross movement of the object in the space, while the parvocellular layer will be responsible for texture and shape and fine details. So you may know this classification as well. The receptive field of the ganglion cells also follow the center surround. So then if we have, this is different bipolar cells when they are stimulating a ganglion cell. So the B and C and D bipolar cells are in the receptive field of the ganglion cell, so that they will cause stimulation of the ganglion cell, causing transfer of the impulse, while the bipolar A and E are outside of the receptive field of this ganglion cell, so that they will not transfer any impulse to this particular ganglion cell. So wide illumination of the retina has a weaker effect than pinpoint illumination. So this is why when you are seeing a diffuse light, you may be not annoyed by the intensity of the light. But if you are if you put a pinpoint light into the eye like a laser, the eye will be very annoyed and will sense the light more, because this light is now stimulating a particular area in the retina, a pinpoint area in the retina, causing stimulation of a single ganglion cell or a group of ganglion cells, and the periphery will not be stimulated. This will lead to sharp edge between the stimulated and the unstimulated retina, because the stimulated retina will be falling on the receptive field, while the periphery will not be falling on the receptive field. This will result in much more augmentation of the effect of the light. So let's say, for example, that this is the receptive field of the ganglion cells, which again follow the on center of surround. Or this, let's take an example of an on center. So there will be a center on and peripheral off, or peripheral off surround. So if there is recording the light recording the electrical potential from this cell, this will be the pattern of electrical potential or electrical impulses. If we stimulate the center of this ganglion cell, so once there is a light on, there will be increasing the electrical charges in this retina, or in this from this ganglion cell. If we stimulate the periphery or stimulate the surround, this will cause inhibition of the visual impulses. So as you can see, the electrical charges will decline as the light here now is hitting the periphery or hitting the off. So it will cause stimulation of the peripheral part of this ganglion cell, which has an inhibitory function, leading to inhibition of the light transmission, as it is evidenced here by decreasing the frequency of the charges. If the light is stimulating or hitting the outside the receptive field of the ganglion cells, there will be no effect. If the whole center is stimulated and the whole periphery is not stimulated, this will lead to the maximum charges or the maximum electrical impulses to be transmitted through this ganglion cell, leading to tonic electrical charges, causing the maximum stimulation. So to summarize, let's say, for example, if we have this is again a graded response, as I told you here. So if this is a small or a weaker stimulus causing a weaker response. This is a stronger stimulus with much more intensity and much more stimulation of the center of the on center of the ganglion cell, causing this tonic response. So if there is a weaker stimulus, this will cause mild hyperpolarization at the level of the photoreceptor or at the level of the cones. This will lead to transmitted to the bipolar, which will cause depolarization. As you can see, this is a depolarization, mild depolarization of the bipolar on bipolar cell, which in case of the ganglion cells will cause, this weak stimulus will cause smaller or milder action potential in the on ganglion cell. The reverse will occur when apply stronger stimulus. Stronger stimulus will cause much hypopolarization, like about minus 60. Sorry, much hyperpolarization in the cones. This will lead to much more depolarization of the on bipolar cells, and the stronger stimulus will cause much more electrical impulses, forming much more action potential at the on ganglion cells. Again, this is the theory of the center surround. We will not go through this again, but there is an if we are if there is an this is an on bipolar and off on bipolar, so there will be a center on and the peripheral off. So stimulation of the center will cause hyperpolarization at the level of the photoreceptors, causing the depolarization of the on bipolar cells and causing full action potential at the on center of surround of the ganglion cells. This picture will illustrate and will summarize all what we have mentioned. Let's say, for example, that you are looking at a stimulus or looking at an object. Let's say, for example, this is a flower. Or let's say it is an apple. Not a very good apple, but it's okay. So let's say that this apple is falling here, or the, let's make this in this example. Let's say that in the dark, or if you are looking at something in the dark, so that there will be a peripheral part which will be or peripheral part of the retina has some light, while in the cone, in the fovea, or the cones of the fovea, the cones are not stimulated by light. So the cones present in the center are not stimulated, but the periphery or the surround is stimulated. So the cones in the fovea are not stimulated. This will lead to depolarization, leading to release of glutamate. In the on center bipolar cells, this will causing a hyperpolarization, leading to inhibition of the on center, leading to inhibition of the ganglion cells, or decreasing the level of action potential at the ganglion cells, leading to inhibition of electrical impulse transmission. And if there is this bipolar, or if this cone is connected to an off-center bipolar cell, this will causing depolarization. Depolarization of the center of this inhibitory bipolar, causing stimulation of the inhibition. This will lead to stimulation of inhibition of the leading to augmentation of inhibition of the ganglion cells, so that this will be again leading to stoppage of impulse passing through the ganglion cells. So there will be no action potential at the level of ganglion cells. In this picture, let's say, for example, that you are looking to an apple, and this apple is falling here. So the apple will be at the center of light, so this will be the stimulus. And the brain or the retina assume that the peripheral part of the apple will fall on a surround, not falling on the center, so that it will inhibit the impulse coming from here, in order to see that this apple has a clear edge. So the apple will be passing through an excitatory cone, and in order for the excitation to occur through the chemical phototransduction, as we mentioned, this will result in the cone hyperpolarization, leading to stoppage of release of glutamate. Then this photoreceptor will be attached to bipolar cells. If it is attached to on bipolar cells, this will lead to stimulation of the on bipolar by depolarization, so that this will result in excitation and transmission of the impulse. If this photoreceptor is connected to an off bipolar cell, then this off bipolar cell will be stimulated again. So how can we stimulate an off bipolar How can we make the impulse be transmitted through an off bipolar? This by inhibition of the center, as the center is inhibitory. So by inhibition of the inhibitory center, this will lead to facilitation of the impulse transmission, so that the impulse will reach the ganglion cell either from the on cell or off cell, by leading to increasing the action potential. So how does the action potential increase? If it is an on cell, this will by stimulation of the center. If there is it is an off cell, this will be inhibition of the center, which is inhibitory in nature. So is anything unclear, or do you understand this? Does anyone have any question? Okay, good. So we come to the final part, or the final destination, in which the impulses leave the retina and enter the brain. So as we know that we have both eyes, every eye has a nasal retina and temporal retina. The fibers from the nasal retina and the temporal retina pass with the optic nerve, so that the temporal retina, the temporal fibers will pass through the optic chiasma to the ipsilateral optic tract, while the nasal fibers from the nasal retina will decussate over the chiasma to contralateral optic tract. So we have two eyes. Each eye will see half of the visual field, but you can see that the left part of the visual field is seen by the temporal retina of the right eye and the nasal retina of the left eye. So both of them can see the left field, and vice versa. The right field can be seen by the temporal retina of the right eye and the nasal retina of the right eye and the temporal retina of the left eye. So in case in the optic chiasma, the decussation occurs, so that the optic tract of the right eye or the right optic tract will carry impulses from the temporal retina of the same eye and the nasal retina of the contralateral eye, and then it will pass through the visual pathway, through the lateral geniculate body and the optic radiation till the visual cortex, until it forms an image at the right hemisphere. This image of the right hemisphere is of the left field, and vice versa. The optic tract of the left eye will carry impulses from the ipsilateral temporal fiber and the decussated nasal fibers, so that they will pass through the left optic tract and the left lateral geniculate body and the left optic radiation and lateral visual cortexes. So they will form an image at the left visual cortex, which is of the right field. So this is the summary of these sentences. So as you can see, each eye only see the monoocular view will be only for the half of the field, not for the full field. So each eye will form only half of the field. The right visual cortex will see the left hemifield, while the left visual cortex will see the right hemifield. This will be the case here. As the fibers will pass, they will reach the lateral geniculate body, which is considered the third order neuron in the visual pathway. And as we mentioned before in the anatomy, it is a nucleus that's formed from 6 layers. The first 2 layers are called the magnocellular layers, and the last 4 layers are called the parvocellular layers. And they have different distribution, or they have a specific configuration of the distribution of the retinal nerve fiber layer, so that the layers 1 and 4 and 6 receive fibers from the contralateral eye, and layers 2 and 3 and 5 receive from the ipsilateral eye. And again, the response or follow the on center of surround organization. Not only the retina, but also the lateral geniculate body receives some connection from the from the reticular formation, and they receive the project mainly to the cerebral cortex, and also they receive a feedback from the cerebral cortex. So this slide has been mentioned and discussed thoroughly in the anatomy, and so I just want to remind you that So let's see what are the afferent axons. If this is the lateral genicular body, what are the fibers that will project into the lateral genicular body? And what are the fibers that will emerge from the lateral genicular body to other sites of the brain? So they receive mainly retinal axons. So the retinal axons coming from the retina will be mainly the main input to the lateral genicular body. They will be divided into the magnocellular and the parvocellular. As the magnocellular, the first two layers, will be large cells that receives input from the Y ganglion cells, as we mentioned, and they will be responsible for detection of the motion in the visual field and flicker. So if there is a stimulus that will be on and off consequently or alternating between on and off or a flicker, then this will be perceived by the magnocellular layer. And the impulses of or the speed of impulse in this visual pathway is very high conduction. Unlikely in case of the parvocellular will be the layer 4 and 5, 3 and 4 and 5 and 6. They will receive from the X cells. They will receive input from the X cells, the ganglion cells, and they will be responsible for receiving the color and texture and depth and shape. So the fine details about the stimulus will be perceived by the X cells in the retina and will be as a second order neuron and will be a relay in the parvocellular layers in the lateral genicular body as a third order neuron. And this pathway has a moderate velocity conduction. Also, there are some areas within the brain that will give stimuli to the lateral genicular body, like visual cortex. Visual cortex will provide the main source for the extraretinal axons and other non-visual sources. They will project mainly to the visual cortex. So area 17 will be, or V1 will be the main area of projection of the lateral genicular body. So the primary visual area, or the area 17 or V1, present at the medial aspect of each occipital loop, and the neurons are arranged to form columns, or sometimes they are called blobs. Leading to formation of 6 layers. The input to V1, this is one, coming mainly, as we said, from the lateral genicular body. And please remember that this input coming from the lateral genicular body will end in layer 4. So we said that it's a 6 layers. The layer 4, specifically layer 4C of Brodmann, this is the final destination of most part or the most bulk of lateral genicular body. So this is the input for the primary visual cortex. The main output, it forms a layer, is from layer 3, goes to the cortical areas, and from layer 4, 5, and layer 6 to the subcortical area. So here we have 6 layers in the visual cortex. They receive mainly from the lateral genicular body, which pass through the layer 4, and the output from the brain or output from the visual cortex will be mainly from layer 3 to the area, to cortical areas in the brain, like for example, like basal ganglia, for example, and from layer 5 and layer 6 to the subcortical area, like the pretectal nucleus. So what are the types of cells in the visual cortex? We have different types, like what's called the simple cells and complex cells and hypercomplex cells. So these cells will be different according to the type of stimuli that they are perceiving. So simple cells are responded to bars of light, lines, and edges, but only when they have particular orientation. So when we are seeing this bar or this line, that we are perceiving by the simple cells that it is a vertical orientation. And when we are seeing this bar, this perceived by our simple cells as it is horizontal. So the simple cells, they will perceive the lines or bars or edges if they have particular orientation. The complex cells will also respond to bars or lines or edges, but especially when they are moving. So we can see this bar by the simple cells, but if we are looking at it like this, or if we notice this movement is noticed by the complex cells of the visual cortex. The hyper complex cells again will respond to the bars and lights and lines and edges to be like the same complex cells. But why they are hyper? Because they are also has the ability to detect the specific length. So when we want to detect what is the length of the target, is it tall, it is medium, or it is short, then this will be the function of the hypercomplex cells. So what is the rule of the area 17? This is responsible for perception of the visual objects or visible objects, but without knowing the meaning of these objects. So we can see anything, or we can know what is its shape or its border, what is its color, what is the details of this image. Is it vertical or horizontal? Is it 3D or 2D? But we don't know what is its name with this area, as this is the rule of the secondary visual area or the secondary visual cortex. So this is area 18 and 19, or V2 and 3, not in the occipital loop, but it's present in the parietal and temporal loop, and this is deal with the complex perception of pattern and forms responsible for object recognition. So we see with area 17, and we know what we are seeing by area 18 and 19. And sometimes there is 10% of cells in the inferotemporal cortex have large complex receptive field and selective for specific stimuli like familiar faces. So when you look to a face that you saw in the past, you will be able to recognize whose face is this. And hence the lesion in this area will cause visual agnosia. So we can see, but we don't understand what we are seeing. Finally, there is the hypothesis of the visual perception. This is called the three-part system theory. So it was the theory or the hypothesis that we can see by three parallel systems that are all integrated into the brain. So the first system is through the retinal ganglion cells, which is the M type or the W cells, which will pass the impulse into the magnocellular layer of layer 1 and 2 of the lateral geniculate body. From the magnocellular impulse reach the layer 4C, the superficial part, in the primary visual cortex, which will, in association with the secondary visual cortex, will be able to interpret the motion of the impulse. So this is called the first system. The second system will be through the parvocellular layers. The P cells of the ganglion cells will be able to see the fine details of the color of the object, like for example, like the color. So if they are perceiving, for example, if you are making this red dot, then this red dot is perceived by your parvocellular cells in the ganglion cell or the X cells in the ganglion cells. This impulse now is transmitted to your parvocellular layers in the lateral geniculate body, which will pass into the layer or relay into the layer 4C in the primary visual cortex into the color blobs. The color blobs here, in association with the secondary visual cortex, will understand that this is a red, and will be able that this is a circular pattern. So this will call the second system. And also the third system will be involved with the P type or the P cells, or that will give impulses to the parvocellular layer from 3 to 6, but will pass through layer 4C into the interblobs, or the areas between the blobs. This will be responsible for the form and shape and texture and stereopsis, and this will be called the third system. So the integration between these different systems is what will enable us to perceive the visual impulse in a holistic approach, or in a holistic manner. So you will be able to detect the color at the same time with the movement, at the same time with the texture and shape, and you will know that if the structure or if the object is in 3D manner or it is 2D. So all this does occur simultaneously without any effort. This is occur because of the integration between these different the integration of these different layers at these different levels, so that you can perceive the visual impulse at the same time. So this is the first picture or the first lecture. Do you have any question in it?